Welcome to Unit 3, Lesson 3, Video Part 2. This part's optional unless you are away. So we're just going to pick up. We've discussed the three types of RNA. Now we need to understand where they come from and what they do. And to understand that, we're going to recall that transcription is part 2 of a 3-part story. As we discussed last day, the 3 parts were you could go from DNA back to DNA, which we call making identical copies or replicas, so it was called replication. Or you could move forward from DNA, you can make mRNA. In fact, you can make other types of RNA like your RNA and tRNA in a process called transcription. So when you use DNA as your blueprint to make mRNA, it's transcription. And then if you further go on and turn your mRNA into building an actual protein, that's part 3 of the story. That's translation, and we'll be discussing that next day. So today our focus is going to be here on the process of transcription. Okay? And to best understand why transcription occurs, it's probably easiest to think of an analogy. So DNA is your original blueprint, your good copy. You don't want to mess it up, you don't want to damage it. So you keep it in the nucleus where it's nice and safe, and it has the instructions for all the possible proteins a cell could make. mRNA is like your working copy, and it's only a copy of exactly the protein you want to build, so just of one single gene. You make a whole bunch of working copies, you send them out to the cytosol or the work site where they're used to build the protein. Okay? So we'll put that down, the analogy. Of the second and third part of the story. Okay, and see if you can follow along and figure it out. So these 2 parts allow the original blueprints which is what molecule is correct, DNA, chromosomes, coding for all the genes, to stay in the safe where they can't be damaged. What part of the cell is that? It's correct, the nucleus. While multiple working copies that would be what molecule? It's correct, mRNA, of a single blueprint. What would a single blueprint represent? So not the entire chromosome, but just a gene. Can be made and sent to the work site, which in this case, the work site of the cell where things are built is the cytosol. well, in particular where proteins are built. to build the house. In this case, the house is the protein. Okay. So that's the analogy of what the process of transcription is. And just this last little part where you build the house, that is translation. Okay. So this is translation. Where all the rest is transcription. And if you were to make copies of all of your blueprints in the body, the entire chromosome, that would be replication. So this is transcription. And just remember, you're getting confused between these words, transcription, translation, replication. Remember the order we learned the processes in, because we learned them in alphabetical order. Okay, so replication first, transcription second, translation last. So before we can do transcription, we have to understand a few things. The cell must know where a gene begins and ends, and what to copy. So if I told you you had to go home and copy the textbook, you wouldn't be very happy with me, to say the least. But the next thing you'd ask is, do we have to do it all? What part of it do we do? The body needs to know the same thing. So to do this beforehand, each gene has its own unique features that help ensure it is properly transcribed into mRNA. Now you kind of have to think about transcription as a party. And for transcription to occur, you have to get people to come to your party, and there's particular people who need to attend if you want a successful party. So the very first step of having a very successful party, we're not just talking, your block party, we're talking your big, like, Hollywood party. You want the A-listers to come to it. So you have this big party and you want all the important people to come. You might hire someone to help you get the party going. And each gene basically has hired someone to get the party going, to get the body to transcribe it. And the person you hire to get the party going, what do you call that? It starts with a P. Who's going to get your party going? Get people to come to it. It's your promoter site. This recruits what are called transcription factors to start transcription. These are like the key people that if they come to the party, everyone else is going to follow. Before the party can actually happen, though, one very important player has to come. So, to start transcription. Because they attract, we could think of it as like the best DJ. They attract what's known as the enzyme RNA polymerase. Without RNA polymerase, transcription will never happen. So first you need to get your transcription factors to recruit people. in particular RNA polymerase. Once RNA polymerase is there, it's got to know, okay, time for the party to start. Let's start copying it, the gene, and it needs to know where to start copying. So, fancy word for beginning or the start point starts with an I. That's right. It's called the initiation site. This is where RNA polymerase actually starts to copy the gene. You don't want a blueprint for one type of house mixed in with the blueprint for another type of house. And finally, RNA polymerase, as much as it likes to party, needs to know when to stop. So there's a fancy word that means to stop or to end life. Arnold Schwarzenegger is a big fan of this one. It's correct. It's called the termination site. It's where the gene ends. It's where RNA polymerase stops copying the gene. So before transcription can begin, any particular gene must have its own unique promoter site, it must have an initiation site, and it must have a termination site. Okay, we're just going to stop the video there and continue in part 3. But if you have any questions about that part, don't hesitate to come see me in class. Otherwise, I hope you have a good day, and I'll see you in class.